Hello and welcome to SciLux, the podcast where we talk about scientific developments and technological changes in Luxembourg, and sometimes also not in Luxembourg, but this time it is in Luxembourg, which means we are proudly powered by Research Luxembourg and recording at the Media Center of the University of Luxembourg. A lot of Luxembourgs here, and even more Luxembourg because we have one researcher based in Luxembourg, one that used to be based in Luxembourg. So you see a full house today in the studio. First of all, we have Professor Elisabeth Letellier, who is the head of the Molecular Disease Mechanisms Group at the Department of Life Sciences and Medicine at the University of Luxembourg. And we have Dr. Mina Tsenkova, postdoctoral researcher at Amgen in California. And the link is that Elisabeth used to be Mina's supervisor. And on top of it, Mina got a very, very prestigious and important prize, Rolf Tarrach Prize last year, or award. I never know if it's prize or award. What's the official award? Okay. Rolf Tarrach Award for the best doctoral thesis. And if you're listening to this episode when it goes live, it means that you have one and only chance to go and see the next prize, which will be exactly the day after. But if not, well, wait for next year. Mina and Elisabeth, thank you so much for coming today.
Thanks a lot for being here.
Yes, thanks for having us.
Whenever I go to a presentation that I really, really like about science, and then it takes me a year to actually schedule the meeting with the scientist, I don't remember anything anymore. I'm not very good at notes because I leave them somewhere. I took a lot of notes when you were presenting, and I don't know where they are. So maybe I should get a bit more organized. But first of all, let's go back to that time. What was your PhD about, Mina?
My PhD was about the role of the gut microbiome in colorectal cancer. And I more specifically was focusing on how diet alters the gut microbiome. And my research led me to zero into the ketogenic diet. And so my project became, how does the ketogenic diet alter the gut microbiome, and how does this affect colorectal cancer development?
Why this diet and not something else?
Diet is actually very important for gut health. And the food that we eat comes into direct contact with the gut and with the microbes that live in the gut. And so it is one of the factors that affects the gut microbiome very importantly.
What I was asking, to be precise, is why ketogenic diet and not something else. But it's nice you actually did a bit of an introduction, because we should do that too. And listeners, if you want to know more about nutrition, actually, that will be an episode that goes live, or maybe is already there. Depends again when you're listening. About nutrition, we'll be talking to another specialist, so you can check it out. But we will definitely discuss diet today as well, because as you said, it's very, very important. But maybe let's start with the basics. So, Elisabeth, we're talking about colorectal cancer. Tell us a little bit about it in general.
Colorectal cancer is a cancer or a mass of cells that start to grow in the colon or in the rectum, depends, and that's why we call them all together colorectal cancer. So it's the third most common cancer type worldwide, and it's actually, in terms of mortality, it's the second cause of mortality among all cancer types. I would say that a few years ago, we were always talking about colorectal cancer being more an disease of the elderly, so 70 plus. Today, unfortunately, we have seen in the latest numbers that the incidence is really rising in the younger population, meaning 50 minus. And that is also why all the governments have now changed, at least in many countries in Europe, have now changed to starting the prevention program at 45 instead of 50.
You already mentioned diet. Layperson perspective. Younger people getting it. It's diet, isn't it?
That's at least one lifestyle factor that we imagine could be a game changer for this early onset colorectal cancer. But there's still a lot of research to be done there. And so we are not yet sure, and we don't have yet really perfect understanding why we have this incidence in early cancer onsets. But nevertheless, environmental factors, diet, obesity, sedentary life, and all these kind of factors do play a role.
I loved your answer because it's a typical scientific answer, right? Because I'm sorry, I'm on the journalist side. So I want, one is two and not some possibly with these and the correlation, causation, all that discussion. But it's good. We have to be safe. And it's important to know that we can't just state easily. Also because it's one of those subjects that I'm sure you're also attacked by on social media everywhere else. It's really discussed a lot. And even when I was actually preparing for this, just out of curiosity, I went out of my scientific and science communication bubble and I went to the more general public bubble. And it's fascinating how gut microbiome and diet in general is discussed in a very much clear path. You eat this, you're going to be healthy, right, Mina?
Yes. That's maybe because the conclusions from studies can be easily misconstrued. And specifically for diet, we're talking a lot about association of specific dietary or lifestyle factors with disease incidence without necessarily talking about causation. And this is something that does sometimes get lost in translation.
So if I get your PhD or your publications, how should I read them then?
You should read them, keeping in mind the various limitations of this kind of study. So we did use one type of ketogenic diet, but ketogenic diets can be very variable and can therefore have variable effects on the gut microbiome and on disease. And of course, one of the bigger limitations of this kind of study is that we are doing it in animals. So this does not directly 100% translate to human disease. And this is something that's very important to keep in mind, especially when selecting the animal model to use.
So we said ketogenic diet a few times, but maybe we should say what it is, because I'm not sure everybody knows. So what kind of food are we talking about?
So the ketogenic diet is a very high-fat, low-carbohydrate diet. Any diet which induces ketosis, which is higher levels of circulating ketone bodies, so that's ketone bodies in the blood, is considered a ketogenic diet. So the exact source of the fats, it doesn't make it a different type of diet, let's say. So it's a high-fat, low-carbohydrate diet with different sources of fats. Historically, in the past, it has been used as a weight loss diet because the absence of sugars from the diet causes the body to break down fat which is stored or the fat which you consume, but this process is much slower and it releases less energy. But there are other benefits which have been observed from this diet, and this is how the ketogenic diet became interesting for different cancer types, not only colorectal cancer, because it was originally thought to reduce the amount of energy which is available to cancer cells to rapidly multiply.
So somebody figured it out by accident? What's the story there?
I'm not quite sure of the exact story behind this, but there have been many different avenues that have been explored, as well as direct effects of the ketone bodies on cancer cells, causing these cancer cells to die, and some other a bit more complex energy limitation factors which have been studied with the ketogenic diet.
Talking diet, we're talking as well colorectal cancer, and we're talking gut microbiome. So before we move on, I think it's a good moment to ask the pub quiz question, because that will also help us set the context.
So to our listeners, do you think that there are more human cells in the body or that there are more bacterial cells in the body?
Perfect. And we have the question. So now we move on. As I said, this whole topic of gut microbiome is a hot topic. So Elisabeth, when you see the discussions, what is the thing that we're all getting wrong, except for you, specialists?
Oh, I don't think that everybody gets it wrong. I think there is raising awareness about what the gut microbiome is. What I feel when I'm giving a lot of talks about the gut microbiome is that it's a kind of a vague term still for the general public. I would say that not everybody knows exactly what it is and what it does. And so what is important to know is that we have these trillions of bacteria, so 100 trillions of bacteria that are in our gut. So slightly more than cells in the human body. And that also means that they have a lot of functions. So they are not there for just being there. They have functions, which means that they are very important for the digestion, of course, but they also regulate our immune system, which is important because that is also what helps us fighting against infections. But it's also important for brain functions. And also we know that it's kind of linked to different diseases. So when it comes to diseases, there is not really something that we can say bad or good bacteria. It's more about something that changes in the composition in the gut microbiome that makes it more associated to a kind of disease. And this is maybe where people also get it a bit wrong, because they believe that maybe a probiotic could change their life, and that's just giving that probiotic could just fix your gut microbiome. But this is a bit more complex than what people think. So there's really some dietary patterns that you need to shape over a longer period. And that's why it's important to do this kind of study that like Mina has really done it over 4 years to really look at exactly the factors that can be shaped and how to use the diet correctly.
I'm asking also because for our listeners in Luxembourg, if you had a chance to go to a different science outreach event, you could walk through a colon, right? And there was, I did it a couple of times, I have to say. I was very excited. And that was also a project that you were part of, right? So how did you decide what to show and what not to show to people?
That's a good question. We have this funding scheme from our national funding agency, which is called PSP, which is for promoting science to the public. So you can get actually just funding support for that. So the idea was, because at that time we were already quite into the gut microbiome, was to really bring this notion of the gut microbiome also to the younger people. So really also students and also like high school students, but also to mix it with making more awareness about the prevention on colorectal cancer. That was why also we asked actually the Fondation Cancer to join that event, but also the ministry actually to also promote the new campaign. And so it was a 3-day event at the Cactus Belle Étoile in Strassen. And the idea was really to walk the people through the gut by understanding at the beginning the function of the gut and the gut microbiome, and then looking at the whole sequence of cancer. And then, of course, also one important aspect is to show the Luxembourgish public also what we are doing as research in Luxembourg.
So it had everything, basically.
Yeah, a bit of everything. And I think it was also important because you have also some of the patients that are coming there to explain them a bit better what is done in Luxembourg. We have, for example, now in 2010, we launched actually a colorectal cancer collection that was established with all hospitals here in Luxembourg. That includes nowadays more than 200 patients. I feel it's important to show also the patients what we are doing and how we can also give back to when they donate samples and also share with us their clinical data. It's important that we also show what is coming out of it.
Difficulty of Luxembourg and data in general, right? 200 for Luxembourg, that's good or bad?
Could be better, but I would say that the advantage of this cohort is that, you know, in many other countries, the cohorts would be larger, for sure. But here, the advantage of this one is that it's small, but it's very well characterized. So we have been able to collect a lot of different sample types, which is not always the case abroad. And it's now really through our collaborations with the Luxembourg Institute of Health as well, we have been able to really characterize it fully. And now it should really become a national cohort that is then available with all the samples, with the clinical data to the whole community.
It's great. So we're talking about sample collection and cohorts and whatever, but coming back to you mentioned an animal model, right? So I'm trying to apply my common sense to the whole thing. Like, how does it work? You put a human colon into a mouse and you study it, or?
So there are many different models that you can use. For my project, we tried to humanize the mouse. So we're talking about engrafting a mouse with donor samples, which were collected in this collection. And then this leads to engraftment of a human microbiome within the mouse. So it is still a mouse gut, but containing human bacteria.
It sounds impossible from my perspective.
It's perfectly possible. Actually, we have a germ-free facility at the university, and this means that the mice are raised in a completely sterile environment. So these mice have no bacteria anywhere, not inside, not on the skin, and so they're raised in this big bubble. And then this allows you to engraft any bacteria that you want without having the competition of a resident gut microbiome, as you would have in a regular mouse, which carries its own microbiome. So this is a huge, huge advantage of our animal facility here at the university.
And that's not common normally?
It's actually not common at all. Yes, it's very expensive. Exactly.
And you always need actually 2 people at the same time, because as you heard from Mina, they are living in a bubble, and even when you manipulate them, you cannot afford any contamination, because then the whole model is useless. So you have to be all the time careful about contaminations. They are living in the bubble. You have to imagine that you basically come in with a whole cosmonaut suit, because you have to be careful about contaminations. And yeah, so it's a lot of work. It's a lot of time, and it costs quite a lot of money.
That's also another point that always interests me when you're doing studies like that is, as far as I understood, there are no holidays, because you basically have to be always there to check on your models, right?
Yes, you do tend to plan holidays around experiments rather than experiments around holidays. But, when you start a PhD, you generally get very, very invested, and it's not the mice, it's my mice. And so, yes, you, of course, need to be very responsible because we are talking about lives.
So once again, what we're talking about here is a certain type of ketogenic diet that you proved to help with healing cancer. Can we say that? With developing less cancer cells, or is it more prevention?
So in this specific animal model, we saw that the ketogenic diet induced cell death. So it's more about killing cancer cells.
And could it also be good for prevention? Or that's not checked yet?
So there are some other studies in other cancer types which show that it can be useful for prevention. But specifically for colorectal cancer, there was actually an article that came out with a slightly different ketogenic diet where they did compare whether it could be useful for prevention or only therapeutically. And they did not see a significant difference when using it in a preventative manner. Does this actually mean that it is not useful for prevention? Not necessarily, because again, there are many different models and there is a lot of fine-tuning to be done on the timing of inducing the cancer and the timing of giving the diet. So this could also be something that simply slips through the cracks of a specific experimental setup.
Is it also related to the stage the cancer is in or not? Are you checking different stages?
With the model that I was using, we don't tend to check the stage. We were looking at tumor size as an indication, but in this animal model, the animals develop more than one tumor. And so then it's very difficult to follow each tumor developing over time to know exactly when it was initiated and how far before or after the diet it actually developed within the colon.
I saw your presentation, I think it was pretty recent, if I'm not mistaken, to one of the institutes in Lorraine when you were talking also about stages and about biomarkers and the importance of actually applying it at certain stage of the cancer to see whether it's developing or not. Because that's also another problem of the colorectal cancer, right? That you can do a surgery and it can be fine, that's it. Or actually it can have a relapse. And it's very difficult to know that it's having a relapse. Am I getting it right?
That's correct. So basically, if you diagnose colorectal cancer in time, you can cure 9 out of 10 cancers, which is amazing.
With a surgery?
With a surgery when you diagnose it in time. Unfortunately, sometimes the symptoms are very difficult to identify, that they are specific to the disease. And so sometimes the stage is already far more advanced. And if it has reached already secondary organs, then the survival chances are dropping, or the overall outcome is far less optimistic. And this is why it's so important to have these prevention campaigns. These are based on the FIT test or on colonoscopy. So the FIT test is actually a test that detects blood in the stools. And this is what you are doing, or what the ministry actually recommends to do every 2 years from 45 onwards. And which is, as I said, based on just simply detection of human blood in your feces samples. If there is just a slightly doubt, or if that comes back positive, then you go to your gastroenterologist. The gastroenterologist is going to do a colonoscopy, and in that colonoscopy is going to identify the polyps or the adenomas, primary tumors, and this is then sent out for biopsy. The LNS in Luxembourg and any other laboratory abroad is then looking at the pathologist is looking at it, and that's how the diagnosis is fixed, so to say. There are a few other biomarkers for diagnosis that are also now in the pipeline, like, for example, microbiome signatures. Because as I said before, what happens in colorectal cancer patients is that there is a shift in the composition of your gut microbiome. So there are certain bacteria that will be less present, others ones that will be overtaking some of the ones that are normally there. And this is what we call dysbiosis. And this kind of microbiome signatures is something that the research field is quite heavily working on.
There was also some protein that you were talking about that is research myosin.
The myosin 5B is a protein that we have have identified, which is reducing expression along colorectal cancer and which could be an important biomarker, prognostic and predictive for stage 2 and the early stage 3, where indeed we have exactly the problem that you stated before. Tumor has been surgically removed, but then after 2-3 years or within the 5 first years, there is a 20% to 30% of relapse. And this kind of 20 to 30% of risk patients, we still have no really good biomarkers that can identify. And so that biomarker that we have identified there seems to be looking that it can actually foresee the survival of a patient, and but also the response to the standard chemotherapies. And this would, of course, be fantastic if we can develop that into the clinic. But as we all know, from the time of the identification in the lab to really being implemented in the clinics is quite a far journey.
Yes, of course. And also it's an interesting journey from the academic world to the biomedical, which I think you are the first guest in my show that has crossed the Rubicon, if I can say, or I don't know. How is it? Is there anything that you would tell people who are maybe considering to drop academia and go work? I don't know how much you can say, right? As well.
I would say that it's been very rewarding. So I would not call it a complete transition into the industry because it is a postdoctoral position. So I am still doing basic research.
You're still publishing as well, right?
Yes. Yes. And so it's kind of in between both worlds, and it gives you a very nice window into what research looks like in the industrial setting. And I have to say that it's very, very, very different, right? It's all about translating fundamental research into something which is applicable. And I think it's very different from what we do in academia. And it's also a completely different mindset. And I've noticed that an academic researcher and an industrial researcher will look at the same presentation and the same data, and they will ask two completely different follow-up questions. So it's a completely different way of looking at the data and what can be extracted from it and how it can be used. Of course, all of the researchers who are in industry, they go through this transition, right? Because almost everyone in this job has gotten a PhD, which you get from the university. So everyone goes through this transition, and it takes some time. It takes some time to change your way of seeing things.
So is there something that you wish you had known before? Or it's too early to say?
No, there's definitely something else that I found very, very interesting. So in molecular disease mechanisms, we focus on colorectal cancer almost exclusively, right? And although I worked on multiple projects during my time as a PhD student, they were all still within the same disease, colorectal cancer. And moving now into the industry, I see that there is much less focus on a single disease. And actually within a single lab and a single team, you can have multiple projects focusing on many different diseases. And so you need to gain broader area of expertise very quickly, as well as the capacity to adapt very quickly and multitask.
Do you think this is a sign that academia, Elisabeth, should be a bit more like that? I'm only asking tricky questions today.
Yeah, that's a tricky one. If you look back at history, many of the big scientific discoveries have been coming still from academia, and they come out because of curiosity of researchers, just looking passionate about their results, about their science. So whereas I agree that there should be a mix between both, I would definitely think that it would be a loss if we forget about this. So there are, as I said, immunotherapy, everything, a lot of these molecular mechanisms that we are talking about, they are coming purely because people were interested in understanding the detailed mechanism of a cell, rather than thinking already at the application of it. And so if we only go for application, we might miss this. And so there are still a lot of things which are uncovered, and so where we need to really invest more time to understand the details. So I feel that there should be a good mix of both.
Don't you ever feel that you would actually prefer to go into a different disease than specialize so much like that?
Yeah, I would say that is what Mina said before is interesting. And of course, if you would have a much bigger lab, that would be great, because I agree with Mina saying that we are not looking at cancer anymore per type, because there are for example, between breast cancer, colorectal cancer, but also between melanoma and there are some mutational profiles which actually make it quite similar, and where drugs could also potentially be able to be of benefit in both cancer types. So we should not look at cancer types anymore, but more within each of these cancer types, we should look at these mutational profiles at more this personalized way to look at cancer. But nevertheless, when you have a smaller lab in academia, it's unrealistic to be a specialist and to have all of these models running for all these different cancer types. So I would say, again, depending on the question that you have, you can have a few more models to test your hypothesis. But now having, like in industry, many more, I think this is rather challenging in academia.
Actually, on this subject, I don't know if it's out of your expertise or not. If yes, please tell me. But if you look at the traditional chemotherapy, how much is it adjusted to the type of cancer? Because from my perspective, again, you just get chemo. You don't really know whether this is adjusted.
No, so there are quite some different regimen for chemotherapy, depending on the stage. For example, for colorectal cancer, you would choose the one or another one, depending in if you are more advanced. And usually at the start, you have often monotherapy, so only one drug is used, but then in a later stage, you can also have a combination. So in colorectal cancer, there are also now many more that try to combine also different therapies, like, for example, chemo with targeted therapy, but also having maybe try starting with one therapy going to another. So there are very different schemes. I would say that chemotherapy is still very useful in colorectal cancer, not for all cancer types, but there are also patients in an advanced stage that will not benefit from it. And the really challenge right now is to identify those patients, because of course chemotherapy, as we all know, are also going to have an effect on non-cancer cells, and that's why it has a lot of side effects. And also for the patient, of course, it's having an effect on lifestyle and also on their daily life. And that's why it's super important to better identify the patients that will not benefit from it. So.
And it circles back to what you were saying about the biomarkers, right? If you're able to know earlier, then at least you don't apply it when it won't work.
Yeah. And also I have to also add that sometimes chemotherapy, because of putting a pressure on the cancer cells, can actually also drive some kind of resistance. So that's why also it is important to identify those that will benefit and those that will not benefit.
Okay, going back to your situation, Mina, and having to specialize in all the possible fields. So after the PhD, what are you doing now? How much can you tell us actually what you're doing at Amgen right now?
So I could tell you a little bit about my primary postdoctoral project, because it is more fundamental research than it is translational. So I moved out of the cancer fields, and I currently am leading a project in the cardiovascular field. And I'm focusing on the role of a specific subtype of immune cells in atherosclerosis, which is essentially the formation of plaque in the blood vessels.
So that sounds to me very far away from your PhD.
Yes and no. So I actually come from an immunology background. And an immunology background actually allows you to work in many different biomedical fields. So before I came to Elisabeth for my PhD in colorectal cancer, I actually worked for a little bit over a year and a half in a virology lab where I worked on HIV. So I've actually gone through a few different fields, but always focused on the immune responses underlying these diseases. So although it doesn't sound close, for me, it still is.
Is it for you as well, Elisabeth, or not?
Yeah, I think so, because the interaction between immune cells, diet, and cancer cells is very important. And it's true that since the beginning, Mina has always focused on this interaction. And so she was always super interested in looking at the immune cells. And that is also what she has published. So sounds quite different, but at the end, the basics of immunology would be the same.
Yeah, you're going for coffee afterwards, so you can already discussed that I'm sure there'll be a lot of questions that probably too difficult for the listener. So let's not go too deep, but questions is the subject I want to move to, because when I was watching one of the videos where you were talking about science, you were saying something, Mina, about the fact that what you love about science is that, you answer one question and you get 10 more. So let's slowly finish with that. What are the 10 more questions that you got recently? Okay, you don't have to give 10, but maybe one that you're on the quest. And this is going to be for you, Elisabeth, as well. I'm giving you more time to think of that.
So this is actually a very interesting question, because it's one that typically you get asked at your PhD defense is, how would you continue your work? What are your next questions? So my next questions at the time were, what are other bacterial metabolites which could have an effect in colorectal cancer, and what are the other bacterial species which could be producing these metabolites? So I was more angled towards fundamental questions. And so it's very funny coming back to this question a year later, and with the experience that I have in the industry, my question would more absolutely be, how can we leverage now this to make a difference? And I don't know the answer to that question.
That's what we said. There's not supposed to be an answer. It's still a question, right? Otherwise, yes, okay. So hopefully in a few years' time, you will not have a different question, but maybe an answer to the one you're posing right now.
Yes,
what do you think?
That is what we work towards every day.
Exactly. What about you, Elisabeth?
I think what I found really interesting in Mina's study is that when we did some different experiments, we realized that you have to have probably an inherent gut microbiome that is shaped in a certain way to be able to respond to the diet. And this is a concept that I think is very interesting, and maybe is also the next step for us as researchers to understand, because if you want really to bring dietary guidelines for cancer patients, we need to understand this first step much better. And so it's a bit like, for epileptic kids, ketogenic diet is used in the clinics because it tends to lower their seizures. And there also we have kind of the idea that there is a responder, non-responder phenotype. And so here in our study, in Mina's study, she saw a bit the same when you look at the results. And this is my next step would be to better understand and help stratifying the patients so that we have a better possibility to transfer into the clinics, because coming back to the biomarker, it's exactly the same. If you put all the patients together, you will probably not see an effect. And so you have to better understand the different groups of patients, whether they are going to respond or not. And then you can do your clinical translation much more efficiently.
I'm impressed that you're only giving one question, because, as a PhD thesis supervisor, PI, and whatever, you get more questions, I would assume, that others come to you with.
Yeah, true. I mean, there are many, many more. The second one is, of course, what you asked before about ketogenic diet. So what I found also fascinating from Mina's studies is that, you asked the question whether prevention would be an option. And so for me, I'm clearly saying no, that's not an option. We also see it from clinical trials that cancer patients cannot cope with ketogenic diet over a long term because it's just too harsh on the body. And so what I found interesting from Mina's studies is that you saw that actually a short intervention of 2-3 weeks of diet can actually reshape your gut microbiome and can have long-lasting effects. And so that is super interesting, because that would mean that you can, by just short interventions actually, and this is coming back to fasting, this is coming back to all what we see right now, and coming back from the biggest oncology conference in Europe. There is much more now going on into this, but it's something where we still need to discover so much. So that would be the first thing, would be to identify this microbiome responder phenotype. The second one is to see what is the impact on the later stages, because Mina worked mostly on a stage that really has the early stages. The next stage would be, and then of course, other diets, like you said before, ketogenic diet is just one of them. And basically it shifts from taking glucose as an energy source to fat. But there are many other diets that could be very interesting to understand much better.
For me personally, the question is also how to convince people to switch and change their diets in general, right? Because sometimes when I have researchers in broader health and whatever, there is a moment when we say, okay, what should we do? And then there's always diet is there, right? Exercise and diet. And it's just, unfortunately, this human nature that we want a pill. We don't want to hear, oh, you should eat healthier, right, Mina?
Yes, that is the case, which is, I think, why raising awareness about this kind of thing is so important. And this, of course, comes back to events like the inflatable colon event, which attracts a lot of attention to this subject and also raises awareness in the younger population.
Exactly. So what remains is to solve the quiz question, because of course that's also raising awareness a little bit, because it makes us think. I think, Elisabeth, you actually solved the question at a certain moment. We were doing poker faces with Mina, but this is also funny, you're not the first guest who solves the question. It's just somehow, when you hear something, it just makes sense to then mention the answer. So listeners, now we are checking if you were listening really attentively to get the answer. But anyway, we're going to solve it. So Mina, if you can remind us what the question was and then give us the answer.
So the question was, does the human body contain more human cells or more bacteria? And Elisabeth did indeed give the answer very subtly, but perhaps she can repeat it for us.
So yes, indeed, we have more than 100 trillions of bacteria in our human gut, so slightly more than actually cells in our body, which is quite amazing and which is something that people often forget. And that's why also it's important to understand that the gut microbiome is really part of our human health. It's really part of our body, and it's a compartment or an entity that we should not forget, and that we only actually understood like 10 years ago that it could have much more impact than we initially thought.
But the challenge is also the fact that it's not so easy to extrapolate the data and say, we do this and all the microbiome will be like that, right? We're moving also to personalized medicine in general, aren't we?
Yes.
Yes, we are.
Absolutely correct. Like probably in a few years, we would talk about the personalized microbiome research, yeah.
Yeah, okay. So in a few years, let's meet in a few years. We'll see what are the questions and what we'll be talking about. And what reminds us to thank you very much for today, for sharing, as I always say, a small part of your world. And listeners, if you're interested, we will share all the links in the show notes so you can read up more. Thank you, Mina. Thank you, Elisabeth.
Thank you so much.
And this is it for today. Don't forget to subscribe, to follow us. We are present on all the social media. You can also suggest guests. We are also on Patreon if you want to support us. And we are waiting for your comments on this episode, other episodes. Remember, we're also on podcasting platforms. This was Silex, and my name is Hanna Siemaszko.